Yep.
Thanks everyone for coming. I've been given the job of introducing Arthur Georges, who's today's speaker, possibly because I've known him the longest, which could be a bad thing. But I promised him I won't do any, won't give away any bad secrets. And he'll be super familiar to many of you, but for those who may not know him, then I'll provide a bit of background information to Arthur. Arthur came to what was then the Canberra College of Advanced Education in 1903. Oh, sorry, that's typo, 1983. And yes, that means he's been here for close to 40 years. He was one of the pioneers and drivers of environmental science at UC, and has been a key figure for that alone. He came here from UQ, where he'd done an honours degree in mathematics before switching to biology and doing a PhD on the ecology and physiology of turtles and the lakes of Fraser Island. And since arriving at UC, he's carved out a research career in turtle biology, ecology and systematics, overseeing a wide-ranging field program in the Murray-Darling Basin, the Daly River, Arnhem Land and Papua New Guinea, to name but a few, often working with Indigenous people. More recently, his focus has been on sex determination in reptiles and on genomics and evolution more broadly, leading to the sequencing of the central bearded dragon genome and many other reptile genomes through the Australian Reptile Genome Program. Today, Arthur's going to talk about the topic that's been his most recent focus, which is sex determination, particularly epigenetics of sex in dragons. And he's promising us ominously 4 more years of fun. Thanks, Arthur.
Thanks very much, Steve. And yeah, hello everyone. Hello strangers, because I haven't been in because I've been avoiding COVID. And I've successfully avoided COVID, so I only come in every now and again. We just got a 4 years extra funding from the Australian Research Council to study this topic of epigenetics of sex in the dragons. And you might wonder why on earth the ARC would be interested in that. But it turns out that the molecular mechanisms behind our bodily functions and development are very similar in this area to the mechanisms that operate in the dragon. And so there's a lot of crossover between what we discover in the dragon and what's relevant to human development and the development of sexual fate in humans. But more than that, some of these mechanisms are so ancient and so pervasive that they are also involved in cancer. And so there's a lot of interest from across the board in what we're doing, and we're drawing heavily from the work that's being progressing under human studies. And I realised when I went to prepare this that I'd given the same talk in October last year before we got the grant. And so I'm not going to give the same talk again, which was the epigenetics of thermolabile sex in the dragon. What I'm going to do this time, if I can get this to work. Oh. Hang on. There we go. So I talked about the epigenetics of thermolabile sex last year. What I'm going to talk about in this is the genetics and epigenetics of genotypic sex in the dragon. So how the sex is determined by sex chromosomes in the dragon, as opposed to how sex is or how sex is overridden by temperature, which was what was in the last seminar. Okay, so I'd just like to acknowledge the large number of people who have been involved in this over the years. And so I'm a bit of a fraud in a sense that I'm presenting work that's been from the efforts of a large number of people working in the lab and in the field. And I've sort of been more of an orchestrator. And so, there's an awful lot of people who have been involved in this over the years, all listed there from various institutions, a lot from the Institute of Applied Ecology, but also from CSIRO, the Garvan Institute, University of New South Wales, ANU, BGI, the Beijing Genomics Institute, the Chinese Academy of Sciences. We've had a gaggle of grad students that have come through this program and worked on really exciting projects. And of course, we mustn't forget the people who look after the lizards in the animal house. Now, the current crop under the renewed funding is reduced somewhat. So we have basically the current crops, myself, Jenny Marshall-Graves and Sarah Whiteley at the University of Canberra. We've got Claire Hollerley at CSIRO, still as a CI on the project. Ira Devison and Andre Martin-Rees, who's a postdoctoral fellow at Garvin's working on Begona. Paul Waters and Nick Lister. Nick Lister's a PDF funded by the grant. Hardip Patel and Udalin over at the ANU, both computational biologists. And at QIMR, Queensland Institute of Medical Research, we've got Sudha Rao and Robert McRae. So grad students so far, we've got 4 grad students now working on the project, but we are recruiting. So we've got one more scholarship up our sleeves, and there's also the scholarship round. So we are looking for more students. Okay, so the topic that I want to talk about is what's the mechanism of sex determination in the dragon, where it's the sex chromosomes that determine the sex of the offspring. And of course, sex determination's it's piqued the interest of the curious and of researchers since the times of Aristotle, and in fact before Aristotle. So in about 475 BC, there was a Greek philosopher who recognized a paternal factor. So it was the first time that they recognized that it was the male that determined, the complement from the male that determined the sex of the offspring. But he thought that boys developed from sperm from the right testicle and girls from the left testicle. So that was the proposition that held sway for a while. And then in 500 BC, Parmenides claimed that the sex of a child is determined by the position of the fetus in the womb. And so they were obviously able to feel the position of the fetus in the womb and came up with some sort of observational correlation that supported that, and that held sway for a while. There was another proposition from Aristotle, which really held sway for a long period of time, and it was the heat and thus viscosity of the sperm during and before intercourse that determined sex. So, you know, if the male ran around a lot and got really hot, and then they got together, then they'd have one sex, whereas if he didn't, they'd have the other sex, presumably. Anyway, sort of from our perspective, sense prevailed when in the late 1800s, early 1900s, chromosomes were discovered, and then sex chromosomes were discovered. So it was seen quite early that the sex chromosomes of a male human are quite different from the sex chromosomes of a female human. We have XX chromosomes in the female, XY chromosomes in the male. So the different sperm carry different complements of the sex chromosomes, and that determines the sex of the offspring. So we're quite clear now. You've got basically through the process of meiosis, you get a halving of the genome. You get gametes that are haploid. From the female, both gametes are carrying the X chromosome. In the male, one's carrying the X, one's carrying the Y. Then when those gametes come together, if there's 2 X's come together, you get a female. If there's an X and a Y come together, then you get a male. And of course, the conclusion from that is that you're going to get a 1:1 sex ratio because of the independent assortment of the chromosomes during meiosis. So this put to bed one interesting proposition that came from Arbuthnot. So back in the 1700s, they went around all the villages in the UK and Europe and recorded the birth sexes of the births across that region, and came up with the first really concrete evidence that we produce males and females in equal proportions. And of course, that's a little bit bizarre because obviously you don't need one male for every female in order to keep the population going. The males would seem to be superfluous, but in the and because the males would seem to be superfluous by logic, this was seen as the hand of God. It was a really good evidence of the hand of God, among the innumerable footsteps of divine providence to be found in the works of nature, there is a very remarkable one to be observed in the exact balance that is maintained between the numbers of men and women. For by this means it is provided that the species may never fail nor perish, since every male may have its female and of a proportionable age. So that was what held sway before sex chromosomes were discovered, and it was realised, science again comes and spoils the beauty of some of these interpretations historically. Okay, so then the question is, well, if you're going to study sex determination, why study reptiles? And reptiles are really interesting because of the diversity of modes of sex determination and developmental reproductive modes and developmental modes. So they're really interesting from that point of view. So you've got parthenogenesis. So you've got obligate parthenogenesis in geckos that arises often through hybridization events that lead to parthenogenic lines, and Craig Moritz would be able to explain all of that side of it to you if you hear one of his seminars in the future. But you've also got obligate parthenogenesis. So if you put a female Komodo dragon in isolation for long enough, they'll throw a fertile clutch of eggs with males in them. And so it's obviously a really interesting way of being able to accommodate dispersal across islands where maybe only one female gets there. And if she is isolated for long enough, she'll throw a clutch of eggs that are not involving a male at all, but produce some males. And you've even got cryptic parthenogenesis, where you're getting clutches of sexual individuals and parthenogens in the one clutch. And that's recently been shown by Lucas Krakovil, our friend from Czechoslovakia. They've also got really diverse reproductive modes. And so you've got this species here, Cyphus aqualis, which lays eggs in one part of its range, is viviparous, live bearing in another part of its range. And then you've got these populations that are intermediate, and some members of the clutch passage through to become born virtually alive, and other members of the clutch are pass through the oviducts as eggs. So quite interesting that you get this transitional form. It's a really interesting area of research by Camilla Whittington, who's one of our colleagues. You also get genetic sex determination in, for example, snakes. They've all got genetic sex determination. And so we say they've got GSD, but in a lot of other reptiles, you've got temperature-dependent sex determination. So in the pig-nosed turtle, for example, the sex of the offspring is determined entirely by temperature. And so that's quite a remarkable trait for a vertebrate. When it was first discovered, it wasn't believed. And in our instance, you get gene-environment interactions, which are really where basically you've got sex chromosomes, but the temperature comes in and overrides the signal of the sex chromosomes and switches the fate. So they're interesting from that point of view, but they're also interesting from a chromosomal point of view. So they're like birds. They've got a few macrochromosomes, a whole stack of little microchromosomes, which is really sort of quite different from mammals. You get XXXY systems like in humans, but you also get ZZZW systems where the female determines the sex of the offspring. And that occurs in birds. It also occurs in quite a few reptiles. A lot of them have heteromorphic sex chromosomes like us, where you can see in the microscope the difference between the X and the Y, but an awful lot of them have chromosomes that are just basically morphologically indistinguishable. You have dosage and dominant systems. So a dominant system is like us, where the SRY gene on the Y chromosome determines the sex of the offspring. In birds, you've got a dominant system where, a dosage system where DMRT1 is present on the X chromosome in double dose. It causes a male. In single dose, it causes a female. And you've got both of those in reptiles as well, those systems. So you've also got different sex determining genes residing on different chromosomes. So not all of the reptiles are going to have the same sex determining gene. And indeed, the sex determining gene appears to be have been reinvented on different chromosomes. So you've got that incredible diversity as well, which makes them interesting and valuable for study. Now, one of the fascinating things about reptiles, and in fact, all non-mammals, is that there's an incredible amount of synteny across the chromosomes. So the chromosomes of reptiles and birds, with some exceptions, are homologous and have been maintained in that state for very many millions of years. In fact, the microchromosomes of the emu are homologous to the chromosomes in Amphioxus. So that's how long these chromosomes have been maintained and the homology of them has been maintained. So you've got all this diversity in the chromosomes on a backdrop of very considerable homology across the reptiles. So that makes them also particularly interesting to study. Whereas if you look in mammals, I mean, mammals have been rearranging their chromosomes all over the place. There's not much homology between the chromosomes of us, for example, and the chromosomes of a shrew. The chromosomes have been totally rejigged. And this is out of a paper we recently published in PNAS, if you want to look it up. So reptiles provide a bonanza of opportunity, basically. That's why I'm saying why study reptiles. So it's a bonanza of opportunity compared with studying birds and mammals. Okay, well, enter our dragon. Our dragon's interesting because it's got sex chromosomes. It's got a 2n equals 32, 10 macrochromosomes, and 22 microchromosomes. And it's a ZZZW system like birds. So it's a female that determines the sex of the offspring. So that's interesting in itself. And we were able to demonstrate this, at least Tariq Azaz was able to demonstrate this using comparative genomic hybridization, where you take the genomes of the basically of the male and chop it up into small probes, paint it onto a chromosome spread, and you do that. You paint the female genome on. You paint the male genome onto the female spread, and lo and behold, the W sticks out like a sore thumb there in green. And once that was done, we could actually show that when you actually looked at the conventional approaches with C-banding, you could actually pick up the sex chromosomes, even though they'd been missed in the earlier studies using the conventional techniques. So a great system. We've got ZZZW sex chromosomes, and the sex chromosomes are microchromosomes, which is why they went unnoticed for so many years. The other thing is that the GSD is overridden by high temperatures. So if you incubate the eggs at low to moderate temperatures, you get a 50-50 sex ratio, just like in humans. That's what you'd expect. But if you start cooking the eggs at higher and higher temperatures, what happens is you get sex reversal of the ZZ chromosomal genotype to a female phenotype. So temperature is overriding the signal of the sex chromosomes. And that makes it really interesting because lots of people have been studying TSD species where sex is determined by temperature alone and there's no sort of clear underlying genetic predisposition. Here we've got a species that's got a really clear underlying genetic predisposition, and temperature comes in and interferes with the programming of development and switches the sexual fate of the XX individuals. So a great model for study. The sex reversal occurs in the wild. So there's a lot of sex reversal in frogs, for example, where they showed sex reversal in the lab, but the conditions required to do the sex reversal didn't ever occur in the wild. So it was considered to be a laboratory artifact. So it was considered that the temperature was disrupting the normal processes that operate in the wild artificially. But in this case, sex reversal occurs in the wild. We were able to develop, Claire Hollerley was able to refine the sex-linked markers and apply them to animals in museums and animals that were caught in the wild to show that the sex reversal actually is a natural phenomenon. And there's quite a few papers there I've listed that go through this in various ways. The sex-reversed animals are larger, they're bolder, and by virtue of their larger size, they're more fecund. So they actually have an advantage in growing larger and being more fecund as a result of the larger body size. Okay, and there's a paper just out by Chris Wild in molecular ecology, which looks at the sort of other selective advantages or disadvantages that might be occurring in the field between sex reversed and normal females. Okay, so our study is basically, the new study is about getting our teeth into the onion to peel away the layers of complexity that are occurring in this sex determination space. So we want to know, how is sex determined genetically in the dragon? And second, by what mechanism does temperature exert its overriding influence? And I spoke about that second one in the last seminar. I'm going to focus on how sex is determined genetically in the dragon. And I just point out that we have sex determining genes in mammals, SRY, we have it in birds, DMRT1, we have a range of sex determining genes in fish, AMH type genes in amphibians, but no one's found one in a reptile yet. There's some candidates, but we don't really know in any reptile how sex is determined genetically. So what we decided to do was hit this problem with a sledgehammer. So we decided that it was critically important to have a genome assembly for this species, and we decided that we would hit the problem of generating a genome for this species with a sledgehammer, so that we could actually not only get a genome sequence for the dragon, but separate out the two haplotypes, and in particular, the two haplotypes of the sex chromosomes. We wanted to separate out the genomic sequence of the Z from the W chromosome. And so to do that, we've hit it with a sledgehammer, so to speak, and I'll run through that a little bit to give you some inkling as to how you go about this. Now, I know a lot of people in this room are not geneticists, so I'm going to spell it out a little bit. So the first technique that we tried was PacBio Hi-Fi. And so with PacBio Hi-Fi, what you do is you take the high molecular weight DNA, you sonicate it to about 20 kb lengths, and then they put these adapters on the end of the get this to work. Yeah, so you put these adapters on the end, which circularize the DNA. They put these adapters on the end, and then they put primers inside the adapters. And then they circularize the DNA, and then they use the primers to PCR around that circle. And they go round and round and round that circle to generate these sort of repetitive sequences for that circle, each with its own level of errors. And then by consensus, they get a Hi-Fi read with 99.9% accuracy. So it's really accurate. And it does it by reading round and round the same piece of DNA repeatedly and coming to a consensus to get the really high fidelity sequence for roughly 15 to 20 kb lengths, which is a lot higher than the old technology, which could only go 150, 250 base pairs. So this is doing 20 kb lengths. And we got some astonishing results. We won an award from basically PacBio Biosciences Asia, about $100,000 worth of sequencing. So they hammered it for us using their technology, and it was in their interest to get a really good result. And they did. We got basically an N50 of 200 megabases. That means half of the genome was represented in contigs more than 200 megabases in length. So really, really good. The maximum was 300 megabases. So a fantastic result. They used a Busco technique where they used sort of known genes to see how well they'd been represented in the assembly. And they got 95% of those genes complete. So they captured basically the whole genome. And there was good evidence that the assembly was telomere to telomere, so it went from one end of the chromosome to the other. So really, really good result using PacBio Hi-Fi. The next thing we did was through the Garvan Institute, used Oxford nanopore long read technology. And what that does is you get high molecular weight DNA. So it can be any length. And then you nick repair it. So if there's any little nicks in one of the strands, you nick repair it, and then you run it through a nanopore, which is basically a membrane with these self-assembling pores. There's 7 units in there, and when you put those units on the membrane, they self-assemble into these pores. And then you have a carrier protein which takes the DNA and carries it to the pore, and the DNA is separated. One strand goes through the pore, and as it goes through the pore, you get a change in the electrical current across the membrane that's characteristic of the bases. And so you get this signal coming out here, which is current intensity, and then using some quite sophisticated technology, they'll actually get the base pairs that were in that sequence coming out. And the really good thing about this is if you've got a, say, 20 megabases sequence, it'll read the whole lot in one read. So these things are ultra long reads. So obviously putting together is going to be a doddle. The downside is that the quality of the reads is not as high as PacBio. So there's a high error rate, and we deal with that by polishing the reads with high fidelity alumina short reads. So that's the second approach we used, and we use that to gap fill the hi-fi assembly. So where the hi-fi assembly had gaps, these long reads were overlaid on them, and it filled the gaps in. And then we use it also to join the hi-fi contigs together into scaffolds. And finally, we use this high-C technique, which is chromatin conformation capture. So basically, the chromosomes in the nucleus, they've got little territories in the nucleus, each chromosome, and the chromosomes interact with each other. And so what you do is you basically you cross-link the DNA. So you basically, where the DNA is in proximity, you cross-link it and bind it together, and then you cut at either end of that bound section, and then you link the ends to make a loop, and then you separate the loop out and cut it into fragments. And what you basically end up with is that's a really crude way of describing it, I've got to say. What you end up with is fragments of DNA where you've got one piece of one chromosome where it's in proximity to the other piece of the other chromosome. So you get these little fragments that you can then sequence through and work out what the association was between the two chromosomes. And of course, because these chromosomes occupy little territories in the nucleus, the frequency of association within a chromosome is much higher than the frequency of association between chromosomes. And so you can actually get a good indication of what's close to what in the genome, and use that to link your reads together from your hi-fi and your Oxford nanopore sequencing. So you use this to link your contigs together into a chromosome length scaffold. So that's how you get chromosome length sequences. So it's really good stuff. You can look this stuff all up on the web. I'm sure you know about Dr. Google. So we did that, and then basically you get these sort of contact matrices which you can overlay the assembly on. So basically, what this is saying is it's showing where the associations are, the close associations are between these fragments in the genome. And because it's linking more frequently in relation to proximity, you get the chromosomes popping out in this density map. So you get this really neat sort of density map, and you you overlay the assembly onto that, and you can see that you've got the macrochromosomes in there. A little bit funny going on there, but anyway, you've got the macrochromosomes there, and then you've got these little microchromosomes here, all showing up as really clear in this contact matrix. So you get a lot of confidence when you get the right number of macrochromosomes and the right number of microchromosomes. There's a bit of manual massaging to go on with this to get it right, but it's a very good visual tool, and you use a program called Juicer where you can actually, manually create this to get the best possible outcomes. Satisfying. Okay, then we were able to use Matt Young's and Janine Deacon's physical map, where they used back probes to actually show, to get anchors on each of the chromosomes, to get little segments of sequence that were found on each of the chromosomes. They were able to anchor that with a technique called FISH. And by using multicolored FISH, they could get anchors that were unique to each chromosome, including the microchromosomes. We could use those anchors then to anchor basically anchor these and say which chromosomes they were, and even down to which microchromosomes they were, including the sex chromosomes. So we were able to use those physical anchors. Matt Young was an honours student here. He did that work, and then Janine carried it further in those two papers that are listed there. So that's called hitting it with a sledgehammer. So where have we come with this? So we've adopted PacBio Hi-Fi as the primary assembly. So we're using that PacBio Hi-Fi to get us a primary assembly. We used Hi-Fi to scaffold the Hi-C to scaffold the Hi-Fi assembly into chromosome length scaffolds. And then we ultimately took the ONT ultra-long reads to read through and hence close any gaps in that. So we actually used the Hi-Fi, scaffolded with a Hi-C, and then we used the ONT long reads to read through the gaps to get a full telomere to telomere assembly for each of the chromosomes. We assigned those chromosomes to the scaffolds using the probes of Deacon and Young. And then we called heterozygous variants, and we're able to phase the two separate haplotypes out using these little variants between the Z and the W. We're able to phase the Z and W out into phased blocks. And then we use the Hi-C reads again to span gaps between those phased out haplotype blocks to get fully phased Z and W haplotypes. So we now have sequences for the Z and the W chromosome, telomere to telomere, with very little error. And so we can now identify what genes are on there, whether the genes on the W are different from the genes on the Z. Is it a dominant system, or is it a dosage system? Is it really subtle, or is it really obvious? So we're right at that stage now where we can start interrogating this. It's really interesting that we located Claire's W-specific PCR amplicon. So she developed a PCR test for sexing these animals. And there's a little amplicon from that PCR. We were able to find out where it was. It's only on the W haplotype. It's right at one end, and there's only one of them, which I think we should write to L et al. And let them know. So that was really good. So, and all the Hi-C read pair mappings to Claire's W amplicon region were linked to the same haplotype. So we had this W-specific amplicon from Claire's work, and we were able to step along the haplotype using Hi-C, and it never went anywhere but onto haplotype 1. So that's the W chromosome. So we found it. So we've cracked the walnut. We haven't smashed it yet, but we've cracked the walnut. Okay. I should just say the three guys up the top there who did this. Like, I didn't do this. These three guys did it, and it's amazing how they managed to pull all this together. Really, really good, especially Andre Martin Rees, who's at the Garvan Institute. They're trying to make their mark in terms of ONT sequencing. They want to become the Australian Centre for ONT sequencing and assembly, and they're really pushing hard to get that reputation. And I believe they're there. That's the place to go if you want to do ONT genome assembly. Okay, so finding the master sex determining gene. So it's not just a matter of finding out what genes are on the W chromosome, what genes are on the Z chromosome. That's the first step. So obviously the sex determining gene's got to be on the Z or W chromosome. If it's anywhere else, like DMRT1, which is a sex determining gene in fish, it's autosomal in the dragon. So it's not the sex determining gene. So the obvious thing is to look along these haplotypes to find candidates for the sex determining gene. And that's something that we had a small section of it. We've now got the whole lot of it, so we'll be able to do that a lot better. We need to look to see which genes are differentially expressed in the ZZ and ZW individuals. So obviously, if it's a sex determining gene, it's going to have to be differentially expressed between the male and the female. And then not only that, because there are a lot of genes that are differentially expressed between male and female, because they're differentially expressed because once the decision's made, as you're developing towards a male or a female, there'll be a myriad of genes that are going to be different between the male and the female. So they're not necessarily sex determining genes. So you have to find which gene is differentially expressed early in embryonic development. And that's a real challenge, but we're sort of getting there. And then finally, a big step is to manipulate the expression of your putative sex determining gene to show that you can reverse sex. And that's something that we're not going to tackle. But who knows? Down the track, someone might give that a shot using CRISPR techniques or some of the other approaches. Okay, so let's have a look at what we've found. So Sarah and Susan have been working on the expression of genes in the embryo. So stage 6 is the stage where you can first identify a consolidated gonad in the embryo. Stage 12 is when it's pretty much making up its decision. It's sort of making up its decision. Stage 9, stage 12, it's pretty making up its decision, and then stage 16, it's fully made its decision as to whether it's a testes or an ovary. So you've got your 3 stages there. Stage 6, 7, stage 12, and stage 16, and basically, Sarah and Susan looked at the gene expression at these 3 stages and looked for differential expression between the male and the female at those 3 stages. And the take-home message here is they found that the genes were differentially expressed by stage 6. So the sex determination is presumably occurring before the gonad is actually consolidated into a discrete entity that can be dissected out. So as an example, there's 2 really important genes. I think it's AMH and DMRT1. Those 2 genes are differentially expressed already by the time you get to stage 6. So what that means is that the sex is being determined very early in the embryo, and certainly before stage 6. So what happened then was that Sarah was able to dissect out the urinogenital tract, which included the gonad tissue. So it's got kidney and gonad mixed in, which is a problem, obviously, for looking at transcriptomics. But we had a look at that and basically found that before stage 4, so at day of lay, stage 2 and stage 4, we looked at those 3 stages, there was no significant differential expression between the sex genes AMH and DMRT1. So we're able to establish, I think, with a fair level of certainty that sex had not been determined up until stage 4, but it had been determined before stage 6. So now we've got a window, stage 4 to stage 6, Sarah is fairly confident she can dissect out gonadal tissue from stage 4. And so we're now going to look at the genes that we found on the Z and W haplotypes. We're going to look at the expression of those from stage 2 to 4 to find out which ones are differentially expressed early in the gonad, and they're likely to be the candidate sex determining genes for the dragon. I hope that's I hope I'm making this simple enough. So all the actions in the stage is 4 to 6 window. And so now we know what resides on 6 chromosomes. We know which ones are differentially expressed in the embryo after stage 6. We've got a means of finding out which genes are expressed earliest, which are likely to be the sex determining genes. And that's up to Yuetzi Yang if we can actually get him in the door, Fiona. So that's up to him to craft a project, a PhD project around this. And so in that sense, lucky him, I guess, or maybe not, because this is tough stuff. But anyway, he'll hopefully be here before the end of the year, and then he'll be able to start tackling some of the questions that I've posed here. Okay, now we do have a gene, NR5A1, which is on the 6 chromosomes. We've already shown that that's the case. How am I going for time? Yeah, okay. So we've got this gene that we think is a good candidate, and we decided that before we did any of this other genomic work, because we know it's on the 6 chromosomes. So Janine showed using fish, probes from NR5A1 painted onto the chromosome spreads, along with a probe that identified the Z and W and found that they were co-located. So we know that NR5A1 is on the 6 chromosomes, and it encodes for a protein called SF1. And SF1 is really important in the sex determination of the vertebrates that have been studied so far. So basically SF1, you've got an initiator like SRY gene, which triggers becoming a male. That initiator then triggers SOX9 to upregulate, and SF1 is involved in that self-regulation of SOX9, that runaway regulation of SOX9, which leads to the upregulation of an AMH gene that leads to a testes. So SF1 is integrally involved in the process of sex determination in the vertebrates that have been studied so far. So we thought this is a great candidate. Let's have a closer look at it. So is this the first sex determining gene for a reptile? Yeah, we were pretty keen. And what we found was there's no difference between NR5A1 on the Z and W chromosomes. We tried very hard to find differences, and we could find no sequence differences between the two copies, one on the Z, one on the W. But what we did find when we looked at the transcriptomics, it is that there's a whole stack of different isoforms, RNA isoforms that have been produced by NR5A1. And in fact, there were basically 3 isoforms produced in the ZZ individual, and 2 of which were functional, I think, and one was dysfunctional because it had stop codons in the middle of it, so it was unlikely to produce a full copy of SF1. Those same 3 isoforms are found in the ZW individual, but there's a whole stack of other isoforms. And so that sort of indicates that there is a difference between the ZZ and ZW in the action of NR5A1. It produces more isoforms of the protein SF1 in the ZW animal than in the ZZ animal. So this is starting to look like, oh, maybe there's something in this, but how does it happen? So I just mentioned that these isoforms are associated with what are called short direct repeats. And short direct repeats are small motifs, 4 or 5 base pairs long, that are repeated at a short distance away in the genome. And what can happen with those short directed repeats is when you come to the post-transcriptional processes, they form a loop and they're excised out. And all of these isoforms in SF1 are associated with a pair of these short direct repeats, which is really interesting. So it looks like what's happening is you've got these short direct repeats that lead to the excising of a chunk of the SF1 transcript that leads to an altered SF1 product. That's really new. We thought it was new. We contacted people in Europe who are specialists in transcriptomics, and it turns out it's not new. Nothing's new. And so if you look in the literature, you'll find examples of this. So that was reported by Yuan Jiang in our second paper in PNAS that came out this year. So you can look that up if you want the details. Okay, so what we think's happening is that you've got basically you've got a Z copy of NR5A1, a W copy of NR5A1. They're very similar to each other in content, but for some reason, the W version's having these chunks excised out. And one mechanism for that is differential chromosomal conformation of the W compared with the Z. And so you get different chunks cut out from a W chromosome than you do from a Z chromosome. And so you get different isoform mixture in the ZW animal than you do in the ZZ animal. And that is what we're thinking is potentially determining the sex. So what happens is basically you get the functional forms coming from the ZNRH5A1, functional forms, they come down here, they associate, because it's a transcription factor, they associate with the DNA in a promoter region of the target gene and switch it on or switch it off, and you end up with a male. In the ZW animal, you get the production of all these truncated forms of the transcript. And what we're arguing is that they associate, those truncated forms associated with the target site and prevent the canonical form from associating. And so they're actually switching off their own functionality. And as a consequence, you get a female. So they're actually inhibiting themselves and you get a female. And that's the proposition that we've put. Of course, it obviously needs testing, but the PNAS liked it. So they can't be all just plausibility. Okay, and so then that brings me to the end. Is there a role for chromosomal conformation more generally? So if we've got chromosomal conformation influencing the differential expression of a gene that's identical in the male and the female, because of differential repeats on the W chromosome compared with the Z chromosome, maybe there's a role for chromosomal conformation more generally, and might it not be thermosensitive? So might not the conformation of the chromosomes in a cell be temperature dependent, and could that be another mechanism by which temperature dependent sex determination occurs? And we've put this proposition to bioassays, and that's in review at the moment. And it's also aim 3 of our ARC grant to examine this proposition. So to finish up, I've talked about the epigenetics of sex in the dragons, but I've really talked about genetics and epigenetics of genotypic sex, not thermolabile sex. And the conclusion, in conclusion, the latest developments in next generation sequencing allow the full phasing of chromosome length haplotypes for sex chromosomes. This is a game changer. This is a game changer in terms of our understanding sex determination. In Pagona, at least, the differences between the Z and the W chromosomes are very subtle, and differential chromosomal conformation may play a role in generating sex-specific transcriptional differences that determine sexual fate. And then finally, chromosomal conformation may be thermosensitive, another avenue by which sexual fate can be determined by environment. And I'll leave it there. Thank you very much. Any questions? Oh, there you go.
You mentioned that at stage 4 to 6, the key time frame to determine sex. The question is, in the temperature override to this mode, what is the key time frame that the temperature will rise? Will that stage 4 stage?
Yeah, so the question is when, stage 4 to 6 is the period when the chromosomal influence comes to bear. When does the temperature come to bear? Basically, we can reverse the sex, I think, up until stage 9. Is that right? Thereabouts. So you can reverse the sex by temperature up to stage 9. After that, it's locked in. And in stage 9, you get ovotestes. So you get gonads that haven't quite made up their mind yet as to which way they're going to go. So it's around stage 9 that it starts to lock in. But of course, you can start influencing it anywhere. And one of the great things about this system is you can incubate them at 28 degrees, which is when the chromosomal sex is operating. And then after 10 days, you can switch half the clutch up to 36 degrees. And so you can actually determine when the temperature is having its effect. You've got full control over that. And so you can factor that into your experiments. So you know exactly when your temperature is having its effect. You can do transcriptomics just before that point and it steps after that point. So it's a very neat experimental model. Yeah.
So the SF1 should be a transcriptional factor.
Yeah.
So in this case, the supporting splice isotopes study should from with isotopes and see whether they have different transcription activities or target region, so that you can have a functional assay to validate the hypothesis.
Yeah, you can. So I guess the difficulty for us is that our next project's focused on thermolabile sex, and we have to bring this part of the work to closure. So Yuan Zhang's still working on some manuscripts, but we don't have the capacity to do more experimental work on it. But it's obviously really exciting and interesting area. It's just that the funding of the current projects on the thermolability and the epigenetics of thermolability. So whether we can, if a student came along that was really interested in looking at that, we'd be able to support them. But it wouldn't be part of the core of our current project. So certainly if a student's interested in pursuing this, then the opportunity's there, the materials are there, there'd be a bit of funding there. So we could pursue it, but that's not a core of the project yet. Yeah, good idea. Craig, you muted, mate.
G'day, Arthur. Can you hear me?
Oh, I'm good. COVID free.
Yeah. Terrific seminar. I really enjoyed it.
Thanks.
So I have a quick. I'm really intrigued by the idea that the chromatin conformation is itself a temperature sensitive. So are there precedents for that in either histone modifications or methylation systems that are thermally sensitive to the point where it actually changes chromatin conformation substantially?
So there's a lot of evidence that both the DNA methylation and histone methylation are influenced by temperature, not directly, but through other molecules that are involved in chromosomal modifications for release of genes for expression and that sort of stuff. So basically KMD6B and JARED2 are two candidates that we're looking at really closely to see the mechanisms by which temperature is captured by the cell, how it's then transduced into a signal that feeds into the epigenetic processes to switch on, to alter the activity of KMD6B and JARED2 in modifying the methylation of histones and releasing and retarding genes from expression, key genes like DMRT1. So for example, we've got a lot of antibodies for those. We're able to manipulate temperature. We're able to use those antibodies to work out which molecules are being modified and right down to localization within the cell to look at that. It's sort of a different seminar. It's sort of the seminar I gave last time, so I haven't included any of that. Whether the chromosomal conformation generally is temperature sensitive is an idea that we put into our RC grant, but there's not a lot of foundation for it. Of course, when cells differentiate into their different functions in the body, chromosomal conformation is a big player in that, in terms of determining cell identity and function. And so the question is, are those processes thermosensitive? Is it a direct interaction of temperature and chromosomal conformation, or is it acting through some of these other chromosomal modified genes like KMD6B and JARED2? So that's what we're exploring, and really we've just put an ideas paper into bioassays, and we've gathered together what foundation for it there is, but there isn't much.
All right, fantastic. Thank you.
Bernd.
Talking of my topic, but
Working with the international expert in this area, Aurora Haruza, forget her name, Haruza, I think, in Autonomous University of Barcelona. She's the expert in this area, and so we're developing projects along these lines. She's sending a student out in September. We're in an early stage of planning. First step is to show that it happens. Second step is to then work out how you characterize it appropriately, and of course, how much temperature do you need? Be an important thing. So it's really pie in the sky. So the RC project's got some concrete things that we know we can deliver on, and then you've got this sort of pie in the sky stuff that would be really risky, but really exciting if it comes off, and the chromosomal conformations in that latter basket. Yeah. That was one last one. One last one.
so is there, you said that the sexual set larger. Is that connected to the temperature and is they have fitness because of it? Or?
It's hard to say. So when you show something in the lab, you have to go in the field to see whether it's an advantage or not. So if you're bolder, maybe you think, oh, you're fitter, you're going to be a better animal, and you're bolder and you're racing around during the daytime, exposing yourself to predation, and so bolder is not fitter under those circumstances. So one would think that laying more eggs because you're bigger is an advantage, but if you don't get to be bigger because you're bolder and you're getting eaten, it doesn't manifest. So one of the things that Chris Wild and Phil Pearson initially were working on was trying to look at how these sort of potential fitness benefits manifested in the field. And Chris was able to show that in terms of their movements and home range, there was no difference between the sex reversed and the non-sex reversed. So you actually don't need a fitness advantage to transition from a ZZZW system to a ZZ system where you throw away the W chromosome. It can just happen through temperature reversing the sexes, and vicious frequency dependent sex determination can drive the W to extinction without any necessary selective advantage in the conventional sense. Yeah. Okay. Okay. Well, thank you very much, everyone.